Any questions in the previous class? And can you see the whiteboard?
One more thing, sir.
Yeah.
So
Yeah.
because these people, they haven't been added, and so what I did was I set up a Google Drive and uploaded the videos on the Google Drive, but I did that only yesterday. So sir, can the
Okay.
junior batch get some time to submit this lab
Yes,
report?
we will give you one more week, but that is it. Because
Yes.
then it is three lab reports, and then I know 3 lab reports becomes a mess. Checking it very difficult. I mean, 150 students, 450 lab reports are not easy. Okay, so please submit it by next week, next Monday.
So for the junior
Okay.
batch, right?
Yes.
Yeah. Thank you, sir.
Thank you. Okay, so any questions? Because today, from the last class, we were talking, last lab practicals, we were talking about production of proteins. Remember, we were talking of production, and once proteins are produced by cells, they have to be also purified. So today, the class will be on purification, because so it's a very strange situation. Production is all about cloning and expression, which is really a molecular biology, recombinant DNA course. Purification of proteins is really on bioseparation. So all these courses tend to overlap. Have you people done bioseparations course already?
We have done
The senior
bioprocess,
batch.
but not bioseparation.
Oh, you've not done bioseparations. Anyway, that's good enough. I mean, we'll talk about purification. And there are two really important things that I want to talk about. One is method and one is analysis. And method and how do you analyze the system. Now, in method, the real part will be talked about by Arunangshu. And the idea is simple that you have to separate the proteins based on various principles, that is essentially properties of proteins. And there are various properties of proteins. Can you mention a few? For example, let us hear you out. Solubility, for example.
Molecular weight of protein.
Size, yes, molecular weight or size, molecular weight, size. What else?
Sir, affinity towards affinity.
एसिडिटी करके तो भाई प्रोटीन एसिडिक और बेसिक तो नहीं होता है. Affinity for what? And then you need to have an affinity, which is an affinity principle. So affinity towards something, of course, but towards what is a big question mark, and how specific. Anything else?
Sir, charge.
Charge, that's a very good one. What is the charge? Is it positively charged or negatively charged? Okay, charge. Anything else? Now we're getting into more complex questions. So these are quite large enough, but you would not have heard of protein structures. But another very interesting point is what is called hydrophobicity. It's very similar to solubility, but we use slightly different concepts because hydrophobic patches are there on the surface of proteins. Ideally, the protein being a large molecule, the internal part consists of proteins which are hydrophobic because they're away from water, and the outer parts contain of hydrophilic parts. So this is hydrophilic because water is here all over. It's all surrounded by water, the protein molecule, and so inside is hydrophobic, right? This is inside is hydrophobic, and outside is usually hydrophilic. But there are always patches of hydrophobicity on the surface, and the degree to which the patches are there on the surface, you have differences in hydrophobicity of a protein. Now this is a good enough number. It covers almost all the stuff that we are talking about, and these properties can be used to separate proteins from each other. Now, what is separation? I mean, I will not talk about these properties and how they are used. This is something which Arunangshu will tell you about. Details, charge. For example, charge separation is done by ion exchange chromatography. Can you well imagine why should it be ion exchange? Because there are two kinds of ion exchangers, anion exchange and cation exchange chromatography. So anion exchange will attract a certain charge, cation exchange will attract another charge. Now think about which charge will be attracted by an ion exchange, anion exchange column versus a cation exchange column. Also think about, and I'm not going to answer these questions, also think about what is the charge in a protein? What is the charge in the protein? So what charge will this attract? Charge which it attracts is the question. And so what should be the charge in the protein which will attract this? And point number two, what is the charge in a protein? And is it not dependent upon the pH? And of course, on the pi of the protein. Remember, at a pH equal to pi, there is no charge. So you should think about what pH and how different pHs would change the charge in the protein depending on what is its pi, and then they'll be respectively attracted to anion or cation exchange columns. So if the pH is greater than pi, will it be attracted to anion? If the pH is less than pi, will it be attracted to anion, or will they be attracted to cation? This is the question. You have a 50% chance of being right, so I don't want you to guess. I want you to think about it. So these are some of the things. Hydrophobicity, for example, is just like ion exchange. We call this hydrophobic interaction chromatography. Solubility, for example, is something which is separated by adding. You remember your Hofmeister series in chemistry? And so what is the top of the cation and anion and things like that? So usually we use ammonium sulfate because it is highly soluble. Of course, you can use other things, but highly soluble, low cost, not very dangerous. So also Hofmeister series, but also very practical concentrations like cost and solubility itself. Size is separated by size exclusion chromatography, and so on and so forth. Size exclusion chromatography. Affinity, there are various affinity tags. One of the most common is the his tag and various other tags. So all these will be talked about later on, but I'm talking about the fact that you use various properties separately to separate proteins. And the real point that one is trying to make here is that in any separation, what are the important factors? There are two things which are very important. One is how much contaminants did you get rid of? How much are you deselecting? How much of the contaminants are you getting rid of contaminants? You have to get rid of contaminants. So it's a very common sense kind of thing. Think about it in a very simple way. Get rid of contaminants. And don't get rid of selecting. Don't get rid of your desired product. So you have to have a system which selects for your desired protein and prevent loss. So it's a very simple principle of separation. Ensure that you select well, that you don't lose your desired stuff, and make sure that you lose the undesired stuff. These are the two opposite flip sides of the coin, right? So in any protein step, and protein purification almost always involves multiple steps, there are two factors which are very important. One, what is the fold purification? And you should learn how to make such tables. What is the fold purification? What happened? Okay, again it's problem. Can you hear me? Oh, no, suddenly. Then it appears back.
Yes, sir.
So what is the fold purification? What does it mean? What is the fold purification? The fold purification is an important term. For example, in your original sample consisting of multiple proteins, literally thousands of proteins, your protein of interest is say 1% or maybe 5%, depends. And the remaining 99% are other proteins. The remaining 95% are other proteins. So now, if I do a purification and I start with 1% protein, and I end up with 7% protein in the final sample, then I have made a fold purification of 7 times. Okay. So 5% protein, starting 5%, will require an approximate 20-fold purification before it gets pure. So you need to have a fold purification which measures how the concentration of your protein increases in the sample. So the fold purification is an important step in purification. Now, a very minor but important point to be mentioned here is that this is very important in the initial stages. It's a very important term in the initial stages. When your protein concentration, and usually before recombinant protein technology came into being, most of the protein purification was done from plants or from seeds or from various natural resources where the desired protein would be 1% or lower. And therefore the required fold purification was 100-fold or more. If the starting protein is 0.5% and somehow you are able to make it 5% in your sample, you have achieved a 10-fold purification. You are nowhere close to purity, but it is a good step, right? If you have start with 0.1% and reach 20%, you have a 200-fold purification. So from a 0.1%, so these are huge fold purification steps. But notice that if you have a 95% protein, pure protein, and you are asked to make it 99%, now, this is a very useful step for therapeutic proteins because 95% may not be acceptable, but 99% is desired. But the fold purification is only 99 by 95, which is say 1.04 or something percent fold. So fold purification is a term which is very commonly used by traditional protein purification people because they are usually working with samples which have very low concentrations of starting concentrations of protein, and therefore fold purification becomes a very important step in enriching the protein in your final purified sample. Are you getting this argument?
Oh, yes, sir.
Yeah. Now the second opposite thing is ensuring or preventing loss, and that is called recovery. And that is a percentage. For example, when you go from 0.1% to 20%, you are doing a 200-fold purification. But you may be losing protein. You may be losing 10% of the protein. You may be losing 20% of the protein. You may be losing 30% of the protein. So your recovery can be 90% or 70% or 80% depending on the situation. What was the total protein now and what was the total protein in the original sample? Are you getting this argument? So the total protein recovered, that's common sense. Now remember, when we report it, we have to think about the numbers, and this is something which Dr. Runangshu will show you. But to be very simple, to put it very simply, for example, one of the things that you would have heard or heard about is called specific activity. Specific activity is the activity that you measure by some assay, and we will talk about activities later on, IU, international units, per milligram of the protein sample. Now, the perfectly pure protein may have an activity of, say, I don't know, depends on from protein to protein, different, let us say 200. Let us say 200 as a random number. 200 IU per mg is the pure protein. So then by definition, you know that if your activity of your sample is 10 IU per milligram of protein, then your protein most probably is 10 by 200. It is 5% pure. Right? And the 95% is other proteins, because your desired protein is contributing 10 units. So your protein, because this is 200 IU per mg and is the real protein activity. So your desired protein in a sample which has an activity of 10 IU per mg is only 5%. Does that make sense?
Yes.
Yeah.
Yes,
so,
sir. Yes, sir.
Therefore, in a very simple way, if I have a 10 IU per mg protein and after purification I measure the activity and I get 50 IU per mg, 50 IU per mg, then by definition I have purified it 5 times, 5-fold. My fold purification is 5-fold. Right? Now suppose,
Yes, sir.
so now suppose the 10 IU per mg protein, there was 100 milligrams of this protein. But now I have only 10 milligrams of protein left. Then you can see that 100 milligrams of protein, 10 IU per mg, the total activity is 100 into 10, which is 1000 units. The total activity. And here it is 50 into 10, so it is 500 units. So you can see that the recovery percentage, that the fold purification was 10-fold, but the recovery was only 50%. So this is how you calculate from the amount of protein that you have and your protein activity. You can calculate both fold purification from the specific activity change, and the change in the amount of protein can help you calculate recovery. Now, did you follow this argument?
Yes, sir.
Yeah. Now tell me, what could possibly go wrong? I have people.
Sir, in what manner?
Okay, I'll tell you. My student came back recently and he said, sir, in this my recovery step, I have recovered 110%. तो मैं थोड़ा गुस्सा हो गया। मैंने सर माप के देखा है सर 110% रिकवरी हुआ, बताइए क्यों हो सकता है?
सर नहीं हो सकता है।
बिल्कुल सही बात है। So I'm just telling you, now think why such simplistic notions do not apply and what could be the possible error in this rather simple description that I have talked about. This is just a question that you need to think about. How can recovery be 110%? What could have happened that recovery became 110%?
Sir, in the process, protein production is happening.
No. Purification कर रहे हैं भाई। आप टॉपिक को मत बदलिए।
Sir, I guess
प्रोडक्शन की बात खत्म हो गई। हाँ।
Sir, I guess it's the error in the instrumentation.
That is one very obvious answer. That is what I told the student, and the student was very angry. This is how you become a poor teacher. I told him it was error. He said, I have done it 3 times. It is always 110%.
No, sir, the set point is changed, so that's why there is an error.
No. These mistakes don't happen. Don't give, assume that the student is intelligent.
Okay, sir.
Don't say that measurement methods में कुछ गलती हो गई, कुछ गलती कर दिया होगा वाला argument तो हमेशा सही है ना. This is a very useful argument for guides. You will never, do a project under me. The moment you come to me with a strange result, say, अरे कुछ गलती कर दिया होगा. That's not a good answer. What is the theoretical possibility? Yeah, go ahead. Think.
Is it possible that the proteins are the desired protein molecules?
Are
They are
you're
ruptured, broken.
You're close, but not too close. No, they don't rupture. Proteins, once they rupture, they don't revert back. It's a very simple answer, really, actually. See, you have 10 IU per mg, which means 5% is your desired protein and 95% is your undesired protein, non-desired, the contaminants. Now, in the contaminants, you can have things which repress the activity. So, actually, you don't have 5%. You are measuring 10 IU per mg, but actually it is much higher than 5%. Because these contaminants actually are preventing the protein from acting as a proper catalyst. Because proteins interact with each other. Okay, and so the answer is rather simple, that actually your activity that you're reporting here is depressed from the real activity that it would have been if it was 5% protein and the remaining 95% was not interacting. This 95% may contain contamination which is toxic or problematic to the protein, which represses the activity. So these are all questions that I will not get into. These are practical questions. Unfortunately, as I said, practicals are being done online. So these are some of the things that you actually see it on your own eyes, then only you can follow it much better. But so the fold purification, these are the two things that you have to handle. And remember that there are multiple steps. So I mean, for example, if there are 10 steps, for example, if there are 10 steps, 10-step purification, then you are in trouble. Why? Because even if it is 90% recovery in each step, what is the final recovery? What would be the final recovery?
0.9
And there are 10 steps into
0.9 2 for 10.
Yes. And this number, I do not know if you have a calculator, I don't have one, would be very, very small. How much is it? Anybody has a calculator? So 0.3 कितना है भाई पता नहीं जो भी हो है ना कोई एक जना को हाँ
so close to 35%.
I was damn good man. I was very good. If I become Shakuntala Devi, the next person, 0.3 bola maine. 0.35. So that's very good. So you see that the percentage recovery is very, very small, 35% recovery. And that's not a desirable figure. So you have to either cut down the steps or you have to increase yields. And increasing yields more than 90% is a tricky business. So they tend to sort of work against each other. I mean, as I said, I mean, very, very beautiful designs. Ammonium sulfate precipitation, for example, you do it in a flask and ammonium sulfate is added. And so, if I add too much of ammonium sulfate, all the proteins will precipitate out. If I add too little, nothing will precipitate out, or some other proteins. So each protein precipitates out at different points of time of different ammonium sulfates. So what do? They do a cutoff. For example, they determine that the protein precipitates out at 50% of ammonium sulfate. Your protein, the protein of interest, the protein of interest, the protein product which you are bothered about, it precipitates at 50%. Then what do? You say, well, first I will add 45% ammonium sulfate or 40% ammonium sulfate and precipitate all proteins which precipitate early. And then after precipitating it out, then I will take the solution. All my protein has got all early proteins have got precipitated out. Then I will remove this medium, and then in this 40%, already 40% ammonium sulfate, I will make it 60% ammonium sulfate. And now my protein will also precipitate out because it precipitates at 50%. So only those proteins will now precipitate which have a solubility such that they don't precipitate at 40, because the ones which precipitate at 40 and lower have already been removed, and those which are highly soluble and don't precipitate even at 60 are also removed. So I have a huge fold purification. My fold purification is very high because I have removed all proteins which are highly insoluble, less than 40% में ही खट से precipitate कर जाते हैं, and very highly soluble. And the ones which are 50% I have not used. Now, the obvious question you will ask, why 40 and 60? Why not 49 and 651? And then I will answer, why not 49.9 and 50.1? Because it doesn't work like that. There is micro mixing, there are local gradients, and precipitation is not exactly 50, maybe it is 50.2, but you know, and so the narrower the range, the more your fold purification will be. But the narrower the range, at 49.9 you will lose some desired protein because it is supposed to be, and at 50.1 some proteins will not precipitate. So the poorer will be the recovery. So the narrower the range you make it, 40 to 60.
In this experiment, what we are trying to do, all kinds of proteins, or we are trying to separate a specific protein?
We are trying to find out one. Sorry. Okay. So now you begin with a mix of proteins, and you of, let us say, A, B, C, D, E, F, G, H till Z, and maybe more, and only X is desired. Out of this AB to Z, only X is the product of interest. And you have found out that X precipitates out, and of course, you will use many properties of X, and I will talk about it one by one. So as a first step, let us say we decide that we will use protein precipitation as a method of purification. Now, protein precipitation is not a method of purification. Protein precipitation is a method of purification only because proteins have different solubilities. So I will use the property of differential solubility to purify the protein. So my protein of interest, I find out it precipitates at 50%. It has a solubility where at 50% of ammonium sulfate, because as I keep adding ammonium sulfate, remember there is, I hope you know the logic of solubility. Sorry, go back one step. As I add ammonium sulfate, things which are less soluble will now, the water will be attracted towards ammonium sulfate, so the water will be drawn towards ammonium sulfate, and the things which are low solubility will tend to precipitate out. So low solubility proteins will start precipitating out at 10%, some more at 20, some more at 30, some more at 40. At 40%, I would have lost a lot of protein as precipitate, because these are the ones which are very low solubility, from 10% to 40% or whatever, 0 to 40%. Then I take this solution very carefully, and then I will add 60% ammonium sulfate. Now all proteins which have solubilities which are higher than 40% equivalent, but not that soluble that they can withstand 60%, will all precipitate out, and your 50%, your protein of interest is included in that number. It precipitates at 50% ammonium sulfate. But all proteins which have solubilities, very high solubilities, and can stay in solution even in the presence of 60% ammonium sulfate, will still stay in solution. So at both steps, you will get rid of highly soluble and very lowly soluble proteins, and your protein will be built up. You have to first find out what is your solubility. मैंने 50 जानबूझ के चुना है. अगर 30 होता तो 20 और 40 लेते और whatever. तो 50, 40 और 60 के कट ऑफ्स में आप एक्चुअली अब प्रेसिपिटेट नहीं कर रहे हैं प्रोटीन को, आप प्यूरिफाई कर रहे हैं. There is a fold purification involved here because you have done 40% and 60%. But why did I do 40 and 60 and not 45 and 55 or 49 and 51? Because I wanted to ensure high recovery. If I had done 45 and 55, even at 45, some protein would have been lost as precipitate of my product X. Some X would have precipitated even at 45, though it is supposed to precipitate at 50. Similarly, not all X would have precipitated at 55, though it is supposed to precipitate at 50. These are the problems of biological systems, variability. And therefore, we will use 40 to 60, compromising on the fold purification, not getting rid of certain impurities, because it's a wide range, but ensuring recovery. If I don't want to ensure recovery, I will use 45, 55 and get better fold purification. So do you see the trade-off, all of you?
Yes, sir. Sir, I have a question. Sir, the question is like, when we mix the proteins in aqueous in water, so do they form a colloidal solution or they form like solution?
They form solution.
Like I want to know about like what is in most of the cases.
All proteins form solutions, never colloids. All proteins form solutions because they have surface charge and interact with water. All proteins are soluble. If they are not soluble, they have misfolded or they are not properly active. Almost all proteins. There are various examples, lipases, etc. I will not talk about them, which are soluble in organic solvents as well. But all proteins, as a general rule, can be dissolved in water. ये रत्नेश जी इसका जोक आपको पता है ना? If you are not part of the solution, you are part of the precipitate. So ठीक है ना? So this is a very old statement about biologists, which may, if you are not part of the solution, you are part of the precipitate. Okay, so all proteins are in solution. They dissolve in water, but not indefinitely. ठीक है ना? There is a maximum solubility index. So 10 milligrams per ml, 20 milligrams per ml. This is about the upper limit. इससे ज्यादा नहीं. 20 grams per liter बस. इसके बाद मुश्किल हो जाए. ये अपर लिमिट है करीबन अप्रोक्सीमेटली. कुछ 30 मिलीग्राम्स होगा, कुछ 10 होगा, कुछ 15 होगा, कुछ 25 होगा. ये मैं एक रैंडम नंबर आपको फेंक रहा हूं. Now, I mean, I need to wrap up because Arunakshu has to take over. I'm taking too much time. Now, the same equivalent recovery versus wo, I will tell you in another way. For example, I ran a column. And you know about migration rates and all that, right? In the column, for example, I'm using size-based methods. And my desired protein comes up at some point in 15 minutes, and all other proteins come, some other contaminant comes in, say, 10 minutes. Or some other contaminant comes in 20 minutes. But it is not so simple really, is it? Because this is how it looks like, and your desired protein comes in 15 minutes like this. But the undesired protein may come like this. Now if it comes like this and this, notice that they are also both very clearly separated. But if it comes like this and this, then you are in trouble. Because you want, this is your protein of interest, this is X. Sorry, I should have written in black. This is X. And this is your contamination, which has come early, which is Y. Now, if I start collecting my X from this point to this point, so that I have 100% recovery, then I'm sorry, you will be collecting a lot of Y as well. So your fold purification will be poor. But if I collect here to here, then I would have perfect purity, maximum fold purification. Fold purification would be very high, but at the cost of recovery, because now this much protein would be lost. So this is the percentage loss. So either you lose this much of protein, recovery is compromised, but your protein is pure, or you collect all the protein but get this much amount of contamination. Because time you are allowed to collect from, so either you collect from this time to this time, or you collect from this time to this time. And if this was also overlapping, same issues here also. So depending on the separation and the spread that is associated with, I now get into Peclet number. आप लोग Peclet number वगैरह याद है? Axial dispersion coefficients, chemical engineering याद है? HETP, ये सब मैं नहीं पढ़ाऊंगा.
We remember the name but not the formula.
Very nice, very nice. मैं बायोलॉजी पढ़ाऊंगा तो कहेंगे सर हम बायोलॉजिस्ट नहीं है सर, अच्छे से पढ़ाइए. मैं केमिकल इंजीनियरिंग पढ़ाऊंगा तो कहेंगे सर हमने वो भूल गया हो. तो ये तो this is a very famous description of biochemical engineer. You know that, na? A good biochemical engineer is one who talks biology in front of chemical engineers and chemical engineering in front of biologists. That is how you become a good biochemical engineer. तो वैसा वाला definition में आप biochemical engineer मत बने तो अच्छा है. ठीक है? So now let us get back. So you realize the trade-off between fold purification and recovery in such a situation. And therefore, there is something called resolution, which Arunakshu will explain to you. This distance is the separation. The separation, and this is the spread. And the spread is this. This is the spread. So if the sum of the spreads, half the spread and half the spread, is less than or than the distance of the peaks, then you have 100% resolution. Okay, spread. So this is a good separation. This is almost reasonable separation, and this is a bad separation. So you need to have
Sir, if the recovery is good, then the width of this middle graph, the black one, this this would be more.
इस केस में कोई टेंशन नहीं है, इस केस में कोई टेंशन नहीं, इसमें 100% रिकवरी, 100% फोल्ड प्यूरिफिकेशन हो जाएगा, मतलब मैक्सिमम फोल्ड प्यूरिफिकेशन हो जाएगा। यहां पे भी कोई दिक्कत नहीं है, यहां पे भी कोई दिक्कत नहीं है, दिक्कत यहां पे आएगी। तब आपको जजमेंट कॉल लेना पड़ेगा कि आप क्या चीज कम करना चाहते हैं, क्या बढ़ाना चाहते हैं। अनफॉर्चुनेटली रियल लाइफ हमेशा ऐसा होता है।
Okay, sir.
है ना? Okay, that is what I'm saying. So let us stop here. And so I'm going to give you a flavor of the fact that there are trade-offs involved in any method of purification. There are trade-offs involved in terms of fold purification and recoveries in every step, and therefore different techniques are used to maximize both recovery and get a pure protein. So let me stop here. And Arunangshu, are you there? I'll call him up on the mobile. So so is it all right? All of you have understood these basic points?
Yes, sir. Sir, I want to know that, sir, if you can please provide some problems on based on this, like
we will provide
you.
you. We'll provide you.
Yes, sir.
Yeah, sure. Arunangshu, you can now take over. I mean, shall I stop my screen sharing? You will, or you can just move in. I will stop screen sharing. Okay. But I think recording should go on. Can you just start your screen? So yeah, भाई रत्नेश जी, सवाल हम एग्जाम में पूछेंगे बहुत कठिन सा जो आप जवाब ना दे पाए, और बाकी समय पूछेंगे नहीं, चाहिए, ठीक है? खुश? Don't worry about it.
No, sir, I am saying that for labs we can have problems, like in this case we are just writing on our own and things. And all those things are theoretical things. So
yes, okay. Arunangshu, can you just make your screen bigger? I mean, I don't know whether I should stop. I think.
Okay, I will start. I have shared it. I will start the presentation now.
No, I don't know why I'm seeing so many screens. Shall I?
Just a second. It will stop.
Can it become bigger? No, you have a small screen, so I don't know.
So can you?
What should I do?
Can you share, see my screen?
Yes, sir.
So
can you? Is it full screen? Can you all of you see full screen?
Yes, sir, we can.
It says
you can see the full screen, not anything else? Okay, fine. That's good. Okay, good. Yeah. Okay. Arunangshu, all yours?
Yeah, sure. So can we start?
Yeah, please go ahead. Please go ahead. Yeah, please continue.
Okay. So last time we told you about about cloning and how DNAs are cloned, and how to read DNA sequences, how to find out the characteristics of a sequence that we have discussed. Now today we are starting about protein purification. Can everybody hear me?
Yes, sir.
Yes, sir, we can hear you.
Okay, just give me 1 second.
Yes, sir. Yes, sir.
So, okay, fine. So let's start about protein purification. So can anybody, so the first feature what we learn about protein in almost all books are there is that first the protein is a catalyst, right? And the second is that.
Sir, I have a few questions from the last lab. If you have time, then you can. Otherwise, you can start this.
Okay, please get it. Please make it short.
Yes, sir. In last class, you were discussing about primer at the T7 promoter. So, what was that primer there? I did not understand what's the use of that primer.
So I was telling about how to figure out a primer to amplify a DNA. So it is, yeah, I was showing on the T7 promoter because that's where usually people make universal primers for sequencing. So you can make primer from any DNA sequences and from whatever region you like, provided that you go through the characteristics of like melting and all those things, right? Is it clear?
So, sir, like why we have to get from primer from like that the sequence was same like it was whatever the sequence was of T7 promoter, the sequence of the primer was also the same.
Yeah, obviously, because the sequence of the primer is that, see, where you have two strands, the 5-3' and the lower 3-5'. So whenever you find a whenever you figure out a primer, so it said it should bind to at least it should bind to one strand, no? So obviously if it binds to one strand, it has to have the sequence of the complementary strand. That's it.
Yes, sir. Sir, the next question is about RBS. So, sir, like I don't know, sir, RBS and Sine Dalgarno sequence, sir. What are these two? Like they are same or different thing?
RBS and Sine Dalgarno sequence, they are separate. They are same thing. The RBS, the Sine Dalgarno sequence specifically talks about the prokaryotic system, and the RBS is that the ribosomal binding sequence. It is actually a universal. It is a sequence which is present in both prokaryotic and eukaryotic system. But in eukaryotic system, we talk that as a Kojak sequence, right? So whenever you are talking about Shine-Dalgarno sequence, you are specifically directing towards a prokaryotic ribosomal binding site. But when you are talking about RBS, you have to specifically mention whether you are talking about a bacterial ribosomal binding site or you really mean the Kojak sequence in the eukaryotic system.
Yes, sir. You shown that in that plasmid the ribosomal binding site. So as we know that Sine Dalgarno sequence is just AGG and AGG, 6 base pairs. But in that RBS, like there were 10 to 11 base pairs, and that were not related to AGG and AGG, and that too like in a primer of like 4 to 5 kb that primer was. So what's the use of that long ribosomal binding site in the plasmid?
So if you try to find out AGG in a E. coli genome, perhaps it will be very difficult to find out that specific sequence all the time. So most of the cases that is called what we can call it as a it is an example that this should be such that. And only RBS doesn't do the job, because what happens is that at the RBS your ribosome gets assembled, right? And then from where it is assembled, it has to start moving towards the direction of synthesizing protein. So before the first start codon is encountered, there is generally a 6 to 18 base pair sequences, long sequence, in which the ribosome actually moves but doesn't start protein synthesis. So that sequence, it has been hypothesized and it has been experimentally shown that that sequence is essential to stabilize the complex before it is ready to put the first methionine in the polypeptide sequence. So that's why you are looking at a 10 or 12 nucleotide long sequences as an RBS. So basically it is an RBS plus a spacer.
Okay, sir. Okay. So, sir, okay, I have a few questions that I will
Now
message you right now. No.
We can have it later. So we have to complete the class also. I think your three of your questions, queries have been answered properly, right?
Yes, sir. Yes.
Okay, so we can have it after the class. So let us first complete the class and then we can have. So okay, so now we are talking about protein purification. So what I said first is that whenever somebody says about a protein, then the very first thing that is mentioned about a protein is that all proteins are catalyst, first of all. So all proteins are catalyst means they cannot start a reaction or they cannot end a reaction. They can only catalyze, make it faster, or they inhibit it or something else. Fine. So the very next point that comes is that the proteins are highly specific compared to our chemical catalyst. Proteins are very specific. So if proteins are very specific, matlab if they bind to only specific substrates or their target sequence, then the obvious question every researcher should ask is that why we need to purify protein, right? Anybody getting it?
So can you repeat?
So what I'm telling is that the property of protein which is mentioned, or the property of enzyme which is mentioned immediately after the fact that it is said as a catalyst, is that proteins are highly specific. That means a protein can act on its substrate despite the fact that there are millions of other molecules in the system or millions of other proteins. Okay, so if this is true, then why we need to purify protein? So that's that any student or researcher should ask about himself, because if we are taking the lysate also, which is a mixture of all protein and metabolite, still it is the fact that the protein will be able to work because it is highly specific. So whenever it gets its substrate, it will work, right?
Yes, sir. But in that case, the probability that it will collide with the substrate will be very less. So we may be wanting to increase that probability.
Exactly. So what we are trying to say is that we need to have a specific concentration of the protein which makes our reaction more feasible than what it is there normally, right? So we want to decrease the background level of noise so that we specifically get more of our target or intended product. So that's one part why protein needs to be purified. But there are also other parts of it, because proteins are not just enzymes, they can be useful for different things. So they are a part of the vaccines, they are a part of different enzymatic reaction, they are immobilized. And so the first part of the protein purification, the first question that needs to be answered, that why protein is purified. So it is very important to say that purification actually leads to loss of a lot amount of protein. So whatever you start, after the whole purification, you just end up with 20 to 30% protein, okay, in the laboratory scale. In the industrial scale, or in a very high very throughput or very advanced scale, you may end up with at most 40% scale. So 60% of your proteins are lost. But still we go for protein purification, because at each and every step of purification, we may be losing a lot of protein, but the specific activity of the protein increases in each and every step. So that is a very important point to remember. Okay. The second point is that we, apart from the applications of protein, we are also interested in understanding what is the protein, what is its structure, how it performs its functions, the mechanisms, and all those things. So that's why we also need a purified protein, because we are not able to do like X-ray crystallography, we are not able to do other like neutron diffraction spectroscopy of a protein to understand its structure and activity functions without purifying it. And the third is that regulatory compliances, is that whenever you are actually having a protein, you need to have the protein as much as pure when you are using it for pharmaceutical and medical applications, because you cannot inject a whole lot of mixture of proteins on a human being or an animal when you are using it as a drug or as the purpose of treatment. So regulatory compliances also makes it important to purify protein. And the third is that and the fourth one is okay, I will talk about the fourth one later, because that is more complicated business. I will come to it.
Sir, I wanted to ask one question, that recovery is proportional to specific activity.
Yeah.
Is that correct?
Yeah. So at each and every step, your protein is getting purified, and you are losing the protein. That means you may be losing the total activity, but the specific activity of your protein is actually increasing at each and every step.
So you said that proteins are catalysts. So are all proteins catalysts? In that case, we will be considering that all proteins are enzymes. So is it the case?
No, all proteins are not catalyst in the all proteins are not catalyst in the sense is that if you consider a chemical reaction in which a protein is involved, you are calling it a catalyst. All right. But when you are considered, there can be different form of catalysis. Some proteins actually they are the part of the control features of the cell. So if you call the control features as a reaction, then it is also a catalyst at that time. But if you think it about only a control, it is performing some control, like the lac operon. So the lac I protein binds to the lac I protein binds to the lac repressor system, and it prevents the activation of the promoter. So whenever there is lactose in the system, it binds to the protein, and the protein gets removed from the promoter, and the now promoter and the RNA polymerase can start its start its function. So in that case, if you are considering that, many people will say, no, this is not a catalyst. Obviously, because it is just a repressor. It is not allowing the promoter to be activated. But many people will consider, no, this is also a catalyst, because if we the rate of removal of the protein will also will actually determine the rate of the activity of the promoter. So that's up to you how you mention and how you look at the functions of the protein. Okay, so can I go to the next slide?
Yes, sir.
So just one last question.
Hmm.
So what is why are we saying that the specific activity of the protein is increasing in these steps of purification?
We are saying the specific activity increasing because the fact is that if your protein is not purified, so the total activity you are getting, and if you divide it with the total amount of protein in the system, so what you get is that at each and every step, this ratio actually improves rather than the total activity. That's what I'm trying to mention. Okay.
Got it, sir. Yes, sir.
Okay, so the next step is that how to purify proteins. So obviously, protein purification is not a very clean procedure, and things has improved with time. But still, I have to add to the note that you people in future may do good research, and you can also come up. But the techniques we are still using in protein purification haven't been so much advanced, unlike the advancement in case of recombinant DNA technology or cloning and all those things. Still, somehow we have to depend on very basic principles of chemistry and analytical techniques to purify protein. So that makes proteins work with proteins a really tidy business, right? So as a part of any purification system, you basically do there are basically three steps. First is that you identify which fraction or from which you have to just purify your protein. Like if you have a mitochondrial protein, or if you have some kind of your protein is a part of endoplasmic reticulum or something, that so you have to enrich that particular portion of the cell in order to purify your protein. So you have to figure out where your protein is actually. If your protein is a part of the membrane, then you have to purify it from the fraction of the cell which contains the membrane debris, right? So you have to figure out from where you want to purify protein. So that is the first part. The second part is obvious, because what you are when you have identified from which fraction you are going to purify protein, then you have to understand that what are the other things that are there in that fraction, and then you have to get rid of them. And the third part is that your protein will have similar structure to at least some other types of proteins. So whatever purification strategies you will be using, so some other proteins will also come out with that come with your protein, because you are using a strategy that is more or less common for both the proteins. Then you have to enrich your protein by removing those traces, and finally when you have done that, you have to do some tests to find out what other proteins are there, and actually how much you have been able to recover. So that's what we call the yield, right?
Sir, I just want to ask a question. Like sir said, like if we purify, then some amount of our desired protein will also get lost.
So that's I am telling it from the very beginning that at each and every purification step, you will be losing some amount of your protein, right? And you will be recovering a portion of the original content of the protein. So that leads to loss of total activity, because if the whole protein was actually recovered, then the activity would have been much higher. So you are losing the total activity, but your specific activity is increasing. That means that you are losing your proteins. And at the very beginning, I said that at the end of the day, after all steps, you will be recovering something like 20 to 30% of the protein. Rest will be gone to rest will be wasted.
Yes, sir. So I was asking
How?
that, the specific activity is equal to enzyme activity upon total protein. So if some of the desired protein is getting lost, so enzyme activity will not remain same. Like if we would have considered a case that the desired protein is not getting lost, there is 0% loss of desired protein, then our enzyme activity would have remained the same. Then it would have been like enzyme activity upon total protein. The total protein would have been decreasing, but enzyme activity would have been remain the same throughout. So it would have implied that specific activity is increasing. But since enzyme activity is also decreasing, because as sir said that some amount of desired protein is getting lost. So enzyme activity is also decreasing, but that decrease may be less than the decrease in the total amount of protein. Therefore, only our specific activity is increasing. Am I right?
So you are asking or you are saying something? Okay, I will listen to your question.
Sir, I am confirming like this concept, like if there would have been 0% loss of the desired protein, then the enzyme activity would have been remain same throughout, and that total protein would have been decreased. So there would have been increase in the specific activity. But sir said that there is a loss of desired protein also. So there is a decrease in enzyme activity. So means the decrease in enzyme activity should be less than the decrease in the total amount of protein. Is it correct?
Actually, I cannot make out what you are talking. So you have to talk to me after the class, and you have to break your statements into individual meaningful modules. If you say everything together, it is not comprehensible, right? So you have to talk. You can talk to me after the class. Just write down your question on the notebook so we can talk about it, right?
Okay, sir. Okay, sir. Okay, sir.
Sir, I have a question. Sir, how do we determine that proteins are being produced in which part of the cell? Is it just the trial and error method? Like how do we do with the genes, or do we have some specific methods to determine in which part the protein is being produced, like in mitochondria or in the cytoplasm, or where? Or if in case of it is being produced in the cytoplasm, then how will you determine? Do you have any methods to determine that, like in which part of the cytoplasm?
Okay, so I will tell you. So I will give you an example with a simple organism called in E. coli. So you know that in eukaryotic cell there are different compartments. There's the mitochondria, the endoplasmic reticulums, and all those things. But a general perception is that in E. coli everything is the cytoplasm itself. But it's actually not true. So in E. coli also we had different fractions, like the periplasmic fraction. So you know the structure of the E. coli cell at least?
Yes, sir.
So what are the three compartments in E. coli cell?
But in E. coli cell compartments, there is cytoplasm, and inside cytoplasm, what can be? There is cytoplasm, and then there is plasma membrane and a cell wall, and inside cytoplasm there is a nucleoid. What can we say now more?
Yeah. So no. So there is actually another membrane outside the cell wall that is called the periplasm. Right. So there are actually two membranes. One is the cytoplasmic membrane, then you have the cell wall, and then there is another membrane that is called the outer membrane. So outer membrane contains outer membranes mainly contain the antigens through which your immune system recognize an E. coli cell, right? So the outer membranes contains antigens like O antigens and all those things. So through which your immune system identifies. So the space between the outer membrane and the cell wall is called the periplasm. So there are techniques in order to send your recombinant protein to the periplasmic space, where there are small crowds of other proteins, and recovery and purification, and thus the recovery and the purification of the proteins which are in periplasm are far more easier. So you can have your protein synthesized with a signal sequence which transfers your protein to the periplasm, and without going into the cytoplasm, you can recover your protein much more easily. So that is one of the projects we are also working on in our lab. Got it?
So the proteins are produced in periplasmic membrane.
No,
Periplasmic.
the proteins are produced in cytoplasm. They are targeted to the periplasm.
Okay, sir. Okay.
Okay, so you can also engineer proteins which you can target to the mitochondrial inner membrane or outer membrane, or you can target it to endoplasmic reticulum. So there are strategies, and there are engineering. Such kind of engineerings are possible, right?
Yes, sir. Okay.
Okay. So now we have moved on to the three important steps. So first is the removal of the cell debris and insolubles. That is obvious. You have to first figure out where your protein is located. Then you have to purify the protein from other proteins in that location. And last of all, some proteins will always come with your protein because they share some similar structure or chemical characteristics. So you have to, at the last step, you have to remove those traces with using different strategies. Okay, so the basic fact is that what are the requirement of purifying proteins? Because now I just want to tell that people used to purify proteins from a hell lot of things, like previously when recombinant protein technology was not there. Yeah, sometimes you have to purify a protein from a tissue. So you start with like 10 kg of bovine tissue, or you start with kgs of tissues, and then you develop your purification strategies, right? And now when we have the recombinant technology over here, what we do is that we simply clone the gene, and the recombinant proteins are expressed. So recombinant protein expression is nothing but to increase the percentage of your protein of interest in the cell uh, uh, through different engineering techniques, compared to what it is there in what it is there normally in the tissues, or in a normal cell, right? So that is recombinant protein production. So for purifying proteins, so we have to start again with different we have to start with different strategies. And first you have to figure out from where you want to purify your protein. So purification of protein through recombinant DNA technology is much more easier and much more cleaner than purifying a protein from like 10 kg or 50 kg of tissue. Okay, but many proteins are uh, uh, very less abundant. If we are talking about the total proteome of the cell, you will see that only 2 to 3% of the protein inside a cell actually represents 99% of the total protein content of the cell, and rest 1 to 2% of the protein are rest of the 3000 4000 proteins. So that is the concentration of most of the proteins of interest inside the cell. So if you are talking about the muscle cells, the actin myosin will consist almost up to 80% to 90% of the total protein of the cell. Rest 10% proteins belong to all other class of proteins. If you look at hemoglobin, then globin proteins are actually 90% of the 99% of the total protein. So that makes protein purification a very tidy business. But whenever we express those proteins in recombinant host like E. coli, then the percentage of that protein fraction increases. But unfortunately, the problem is that many of the proteins which are actually soluble or which are functional in their original tissue or original cells may not may not be active, or may not be in their native state when you produce them in recombinant host. So that makes the recombinant business challenging, and thus what happens is that you have to recover those proteins from those hosts only. There are no other ways. So in order to do that, we have to depend on some basic strategies and basic reagents. So first step of when you start purification of protein is that you have to ensure that the protein there, the protein you want to purify from the cell is actually there. It is not being degraded, because what happens is that when the cell dies, or when the tissue is detached from a living animal, everything starts, a mayhem starts, microorganism starts growing. There are proteases which degrades protein inside the cell. So everything is out of order, and the chance that your protein is surviving can be very less, until and unless you do something in order to protect your protein of interest, right? So the first thing what we have to take into account is that we have to ensure that our protein of interest doesn't go, that is not degraded. So what we do is that we take the tissue, or take the cells, we lyse the cells and add protease. We add chemical substances that uh, prevents protein degradation. Okay, so we call it the PICs, protease inhibitor cocktails. So it consists of pepstatin, aproteinin. There are many parts of it. So you can Google the name PIC and you can have the name of the whole components. Okay, so like pepstatin inhibits the action of proteases like pepsin. Aproteinin also prevents the action of serine proteases. Okay, so the proteases are your enemy when you are doing some kind of protein purification, and you have to stop them from degrading your protein. So we add PICs. So the next important thing is that you need buffers. Okay, because your protein is inside the cell in a cytoplasm which has a completely different composition and characteristics. Now you want your protein to be purified, so you want to have your protein transformed from the cytoplasm or any other fraction from which you are purifying to another solvent system. And now the solvent system of interest is your buffer. So why use buffer? Why not use water? We use buffer because proteins are, they may be acidic, they may be basic, they may be neutral. Okay, and there are so many proteins that when you add them to water, it may change the pH of the water itself. But your protein of interest may have a very small range of pH range in which it is stable, or in which it is in the native form, or which it may be in the folded state, or in folded state. So you have to transfer your protein into buffer. This is one thing why buffers are needed. The second thing is that the protein has a pi. So there is the protein, whenever you have your protein, it has a pi. So if the pi means, there basically the protein has two functional part. One is the the polypeptide backbone, that the amide bonds and the NH groups and the CO groups and all those things, and the side chains. So pH the pH or the acidic or the basic properties that a protein displays is the property of its side chain, not the polypeptide backbone, right? So you have to maintain the protein in such a condition that you want some kind of charge on your protein. Okay, so either your protein may be negatively charged, or it may be positively charged, or it may be it may and if they're in a if they're in a zwitterionic state, so there will be no charge. So that's why we need to have the protein at a particular pH level from which we can purify it. So we implement ion exchange strategies to purify the proteins, for which we need solvent systems which are buffered for the ion exchange process to be carried out. Okay. The third thing is that the protein content.
What is PI, sir?
PI is see, the PI is something like the, see, I will tell you just in a very short way, because see, I have to tell a lot of things if I about to analyze PI. So I think that
yes, just in short.
That's a part of a theoretical class. But what I will say that the PI is something, see, there is a particular pH when your protein has no net charges, right? Getting my
Yes,
point?
sir. Yes, sir. And Yes, sir. I understood. I got it.
Okay. So in that state, your protein is least soluble in the solvent, and it has a propensity to precipitate out because of the hydrophobic interactions it can have with other partners, other molecular partners, because they all have hydrophobic patches. So has if your protein has a particular pi and the pH is above it, it may be either positively charged or negatively charged. So when it is negatively charged, so all other molecules are negatively charged, so they will be repelling each other and there will be no precipitation, so they will be stable in the solution. It is also true if it is positive, but if it is having no charge, so there is a possibility that it will be precipitate out, right?
Yes, sir. Got it. Got it.
Okay. So the next thing is that you have to prevent your protein from being oxidized, because oxygen is something which is available in the atmosphere, and everything has a chance that it get oxidized because of the oxygen in the atmosphere. And there are also disulfide bonds which determine the structure and activity of the protein. So they are sensitive to oxidizing conditions. They are sensitive to oxygen. So we add reagents like DTT, dithioerythritol, or mercaptoethanols, which which prevents the oxidizing, the normal oxidizing process. And also, the our main focus is not to have the disulfide bonds, keep it as much as open, so that when we do some refolding reaction or when we store some proteins, that the native disulfide bonds are recovered rather than the wrong ones. So that's why we avoided or add DTTs and all those things. The third is salt, obviously, because proteins always in the biological cell there is always ionic the solutions are ionic, so obviously you have to maintain some level of ionicity in the solution, and that's why we need salts. We need to add the chelating agents like EDTA, because some polyvalent metal ions tend to form bridges and complexes which can interfere with the protein structure and their stability. And when our protein is not in its native state or folded state, or it is in some kind of aggregate, and or it is some kind of uh, inclusion bodies, so then we need to have this protein open, the protein chains open. So normally it is one of the toughest job in the world in order to keep your protein in a straight chain. The we called it the melted state. Okay, so all proteins are a kind of melted globule. You can think about a soft jelly ball, and it is very difficult. And what happens is that we are actually terrified when this molten globule actually collapses in something like a more compact form, and that we called inclusion bodies. So when we have inclusion bodies or aggregates, we have to take the we have to refold the protein, and in order to refold the protein, we have to take it back to the normal linear state when there is almost no interaction. We call those state as the random coil state. So we have to take the protein into the random coil state, and then we have to again make it fold back to its original state. So denaturing agents urea and guanidinium hydrochloride helps protein get back to their random coil state. Get back to their yeah, right, random coil state from which they can refold back. And lastly, in order to preserve your protein, you need to preserve it at low temperatures, and sometimes icing and crystallization may harm the protein, and even frequent freeze thaw cycles also harm the protein. You need a cryopreserving agent like glycerol, which helps you preserve your protein from long term. So any questions regarding the component I talked about, the reagents I talked about for protein purification?
Sir, do we get like you talked about like denaturation, melting, and then again renaturation. So doesn't it happen many times like the protein is being after folding, when the protein folds, it folds into something different kind of protein. Like folding, if different kind of folding can result into different kind of proteins.
Yeah, obviously. So that's what I actually mentioned at first is that your protein of interest is just a second. Your protein of interest is something which is in a biologically active form, right? But most of the time is that whenever we express these proteins in recombinant host, they may not fold properly. So protein folding is itself a very big subject. There are different kinds of forms of protein that may appear. Like your native state is, see, first of all, the folded protein has a thermodynamic property. Okay, so the all folded proteins are not of our interest, because some folded proteins will be very stable, but they may not be biologically active, right? And another thing is that some proteins tend to aggregate. Okay, so aggregation is something in which many protein molecules come together to form a different a small island in the cytoplasm or in some kind of compartment, which causes some kind of biochemical imbalance in the cell. So you may be hearing of many clinical conditions which occur because of protein aggregates, like Parkinson's disease and all those things. Many proteins tends to aggregate, or so that is also something. And the last is that inclusion bodies, they are the last extreme forms of protein which are very compact. So I told you that the most interesting and most active form of the protein is a molten globule form, in which it is partially melted and partially folded. And basically the structure is basically flexible to perform its function. So we all want that our proteins to be in the molten globule form, rather than in some compact form in which it cannot do any kind of conformational changes to perform its own function or folding or anything, right? So we want our protein mostly in the molten globule form. And it it can form the molten globule form when the polypeptide chain is completely extended, so there is no folding. So that's we call the random coil form. Okay, so in order to have your
Yes, sir.
protein, yeah, tell me.
Sir, I was reading in the news like that by using the AI, now the
No,
AI has the
no,
power
no,
to.
no, 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 no. We are not going into those things. I'm just telling you for your interest. No, that's a part of the research. You cannot quote newspaper readings here, right? So I am just telling that what are the forms of protein that are of interest to us, and what is the use of the reagents in order to achieve those forms to have our particular functions, right? Okay, so if no questions from this part specifically about the buffers or oxidative state or the salts, then I can move on to the equipment part, right? So the next part is that the equipment and the pH meter. So the pH meter is obviously essential because you have to have your pH of the buffers checked all the times. The centrifuge is the most essential part in order to have the fractions of the cells to be separated. The spectrophotometer is always handy in order to see the concentrations and all those things. And the electrophoresis unit is the heart of any protein work in any lab, because you have to separate the proteins on gel and you need the electrophoresis unit. So now coming to that part of the purification, we talk about what are the different strategies to purify protein. So the first strategy that we all love and it is easy is the affinity purification. So what we mean by affinity purification is that your protein of interest has some kind of properties through which it has developed a strong affinity to something. And once this property can be exploited in order to have your protein concentrated in with something, and rest of the proteins will be washed out through simultaneous washing and dilution, and then you can recover your protein. So there are different strategies for affinity purification. Like the most popular and the most useful is the histag. Nowadays, we also have magneto-affinity purification. That is that the histag is bound with a ferromagnetic particle, which can be pulled down using magnetic properties, and your protein get concentrated. So these are these techniques, magneto-affinity techniques, come handy when you are trying to purify your protein from very small amount of culture, like 1 ml culture, right? The second, and the rest of the process, as you, I guess you all know, because these are the fundamental process, fundamental properties and fundamental things that might have that are covered in your theoretical classes. The precipitation, ammonium sulfate precipitation, polyethylene ethylene precipitation, and lastly, most importantly, the organic solvent precipitation, like the ethanol and acetone precipitation. Proteins will precipitate when organic solvents are added to them. And the last and the most important part that we all know and we do most of the times is the chromatography, the ion exchange, size exclusion, affinity, and hydrophobic interaction. So I will talk about one technique today, that is the affinity purification using nickel NTA. So what happens is that the nickel is complexed with sorry, any questions? Okay, so the nickel is complexed with nitroloacetic acid. As you can see the structure over here, it's the nitroloacetic acid, and this is bind to an agarose bead. So what happens is that when you add your protein, which are having histidine tags are nothing but 6 histidine molecules attached to the end terminal of the protein. So when the 6 histidine molecules are there, so they form a complex with the nickel, as you can see after the picture in the binding, after binding. So they form a complex, and these now what happens is that your protein is now bound with the nickel, and the nickel part is bound with the agarose beads. So the agarose beads are small beads which are precipitated, and then you wash the solution. All other proteins which doesn't have an histidine tag will be washed out, and the agarose part will be remaining on the column. And then you put an imidazole solution, which will now replace the binding of the histidine with the nickel, and your protein will be simultaneously eluted. So one thing to note over there is that you see that the side chain of the histidine has an imidazole group. So we are replacing the histidine with a similar molecule which actually resembles the side chain of the histidine, that is imidazole, and at higher concentration, in which the histidine bound protein is released and the imidazole gets bound to the nickel center. And this is one of the examples.
What is the size of this agarose? That agarose. What is the size of that agarose? How small this is that? Like in the size of nanometer, range of nanometer, or something big?
It is just as the agarose beads are size of the dust particles. You can see with naked eye only.
So then the naked, if you can see with naked eye, then it is like 0.1 millimeter or something.
That is not our interest, no. Why should I measure agarose bead?
So that like all the reactions are happening because of that. So why should, like, isn't it like a concern to us?
No, how it is a concern to us when you are purifying a protein?
Okay, sir. Okay.
Okay. So so now when we have our protein is bound to the histidine beads, the nickel beads which are attached to the agarose, we then perform the elution, and our protein of interest is eluted out. So as you can see, the E. coli lysate is the whole cell lysate, and the thick band, the dark band over there is your protein of interest. And then when you put the nickel NTA beads, the picture of the nickel NTA beads is there. You can see the light green, bluish green beads over there. So when you put, your protein is now bound, and this is the flow through. What has flowed through the beads, that fraction you have run we have run on the gel, and then we have washed the column with our wash buffer. So the binding buffer contains no. The binding buffer means initially the protein is in your binding buffer, which you add to the agarose beads. So the protein, the histidine tagged protein now gets bind to the nickel center of the agarose beads. Then you wash it with 5 millimolar imidazole, So the 5 millimolar imidazole is added in order to ensure that all other impurities which are loosely bound to the nickel centers are washed out. And then you finally add 20 millimolar imidazole to the bounded protein, and your protein is being eluted. So after that, we do dialysis in order to remove the imidazole from the solution, because imidazole is generally toxic. And that's how we achieve the purification of protein using affinity. And then we have to run all our fractions on SDS page. What the picture I have shown you before is a picture of the SDS page. So SDS page is the heart of any purification system that we do. And for all the fractions we get, like the first E. coli lysate is the whole fraction which contains your protein. The second is the flow through, the the part which has come down from the beads after your protein is bound. The third fraction is the wash fraction, the buffer you have used to wash the beads. And the third is the elution fraction. So each and every time you do a protein purification, uh, after every step you have to run all the fractions you have obtained, starting from what you have added, what have flowed through, what have washed, and what have eluted. So that gives you the correct idea that what is happening to your protein. Many a times, yeah, the as you can see, the flow through is very clean. Many a times it doesn't happen because your protein might not be getting attached to the nickel center. And if you don't run all the fractions, what you will end up is that you will say after the elution, no, I don't have, my protein has not been purified. But you have to find out where you have lost your protein. So all running all fractions on the gel is very, very important. And then you have to figure out that where you have lost your protein. So despite all the methodologies and all the techniques, one thing I can tell you very surely, that every time you do a protein purification, it is a new experiment. What you have done previously with the same protein may not work, and you have to change some things each and every time to have your protein purified. So even though the practices and the procedures are very standard, it is an experimental procedure that you have to do every time, and you have to look at every fractions and every minute details whenever you are purifying a protein to have the maximum recovery. So now the SDS page. So I will let's just I'm not going to tell much about the SDS page. You can ask me questions about the SDS page. Have you any idea what an SDS page is?
Yes, sir. We have learned that in
Okay. So acha, there are many people in the class. Why everybody other, I think nobody is interacting. Can anybody hear?
Yes, sir, you're audible.
Yes, sir.
Okay, so yeah, you guys, nobody has any questions regarding the previous slides? You can ask me, not a problem. Plus, keep it small and don't go into business which are not a part of here. I can answer those questions also, but that will not be fruitful to your current perspective of study, right? That's why I'm not answering. I know the AI system, the AlphaFold that has come, that is an AI system, the AlphaFold by Google. So these are techniques which are being used today to study protein folding. But obviously, like, you have to learn something more in order to go at that level, because you have to know many things when you are trying to do those experiments, right? So any other questions from the previous, any other students?
Not as of yet, sir. If we have it, we'll ask at the end of the class, maybe.
Okay.
Yes, sir. We will ask all our doubts at the end of the class.
Okay, so I will not go to the SDS page thing. Actually, yeah, this is taught in every classes, and you all know. So maybe I will ask some questions now so that I can understand that you have got. So uh, what is the use of SDS in SDS page? Anybody?
Imparting.
Sulfide radical sulfate, and it SGS, it is a detergent, and it is used to denature the proteins from its tertiary structures to its primary structures, so that because the proteins have different shapes, so that all the proteins can come in the same shape.
Anybody else?
Sir, before imparting the charge.
Sorry, once more, please.
For imparting charges to proteins.
Right. So what charge?
Negative
Sir,
charge.
I do remember.
Okay. So it gives a uniform negative charge to the polypeptides, right?
Yes, sir.
Fine. Okay. So again, what is the function of APS? Anybody? What is the function of timid?
So catalyzing the polymerization process.
What type of catalysis is this?
So using like ammonium persulfate is used as the oxidizing agent, right? And free radical based, like to proceed by the free radical based polymerization.
Yeah. So timid is your free radical initiator, and ammonium persulfate helps in propagation of the free radical chain reaction, right?
Yes, sir.
Okay. And why use stacking and resolving gels separately?
So that all the proteins come at one place. Otherwise, they will be what is called dispersed throughout the gel, and we will not get a visible like clear spectrum.
Yeah. In order to ensure maximum efficacy of separation, you have to all the proteins should come to a single band, and then after that, in the resolving, they get separated. That is the whole idea. Nice. So obviously, so I will end up here and take up questions, and you can.
Sir, so can I ask doubts?
Just a thing to add. Try to be topic specific. Don't go very broad, because that is not good for you and me both. Carry on.
Sir, can you open the whiteboard, because the concept I want to ask, like, we means it can be only means understood means if. Sir, can you open the whiteboard?
I don't think I have that luxury of opening whiteboard, because I am through internet, not the.
Okay, sir, no problem. Sir, like, if the recovery is 100%, so enzymatic activity would be 100%.
How do you connect the two things, that enzyme activity and recovery? How you are connecting?
Sir, what is recovery? Recovery is our desired protein. Is that right?
Yeah.
Okay, so recovery is our desired protein. So if our desired protein is 100%, then what is enzymatic activity? Enzymatic activity is the activity of the desired protein. So enzymatic activity of that desired protein would be 100% if that desired protein is 100%.
Now I told you in
Sir, how do we define this enzymatic activity?
Just a second. What I told you through the class is that you say you have recovered 100% of your protein, but
yes,
who has confirmed you that all your recovered protein are in native state, which is mandatory for enzymatic activity?
No, sir. What I'm saying that like if there is a protein, desired protein, whatever protein we have, we know, like, suppose its enzymatic activity is 200 nu per mg, for example, and after the after whole purification, if our recovery is 100%, after whole purification, suppose if our recovery is 100%, then our enzymatic activity would remain 200 nu per mg. Is that right?
It may be right, it may not be right, it may also increase.
No, sir. specific activity increases, not enzymatic. Specific activity is equal to enzymatic activity upon total protein. I'm just saying about enzymatic activity. Like if the protein, desired protein which we have, we get after purification the desired protein 100%, then the enzymatic activity would remain 200 nu per mg, which we uh, which we which we stated.
Sir, in that case, like enzymatic activity will not be like mg per mg. Like a specific activity is per mg, but and I don't think that enzymatic activity will be per mg.
So I think this is a question that you can best refer to your theoretical classes, because I first of all I am not getting the questions very properly, and I am not a person who is of that like uh, of this very specific mathematical and this part. So I think you can ask it in your theoretical class more frankly, because they will be able to answer you more in a better way than me.
Okay, sir. Okay, sir.
Okay, so I can recommend you some books, like you can go through Trevor Palmer in order to find out these things of interest. You can read Trevor Palmer, one thing. You can enzyme catalysis and all those things. This part is very good, but that is a book which is specifically dedicated to enzymes. So you must be getting all your queries
What is the
answered.
name of the book?
Trevor Palmer.
Name of the book?
Name of the you just put Trevor Palmer. I will. I think Introduction to Enzymology. So you see this book?
Yes, sir.
So it contains all the mathematical question on enzyme kinetics.
Again, you are asking something which is I don't understand what does it mean by all mathematical, but obviously most of your queries are going to be you will get all the Michaelis-Menten, Landweber-Burke plots and all those things. Most of things are covered, obviously. Okay, any other question? Anybody there? Any other questions?
No, sir. Thank you, sir.
Okay, so if I
have a question.
Please let me know. Any other
Sir,
question?
sir, you told that actin and myosin makes 95% or something like that in normal cells. Sir, so sir,
I said
just these
muscle
two
cells.
proteins.
I said muscle cells,
What?
not normal cells.
Okay, sir. So, sir, do we ever try to separate them also?
No, I didn't. I didn't purify actin myosin. Most of the cells, depending on the tissue they are there, they have certain amount of the proteome which is enriched. Like I told you, in muscle cells, most of it is actin myosin. In red blood cells, it will be the globin proteins which are involved in hemoglobin. So that's the calculation.
Sir, like actin myosins are like these are not like protein molecules. Like I think these are polymers, right?
What is a protein?
Oh no, sir. I mean protein of like after three-dimensional folding, then also it polymerizes, I guess.
Who said you this, yeah? What is a polymer?
Oh, polymers are made of many monomers.
So there are homopolymers, there are heteropolymers. So your polyethylene is a homopolymer where there is a single repeating unit which builds up the whole polymer. So protein is also a polymer in which there are 20 different units. Your DNA is a polymer in which there are 4 different units. That's it.
Yes, sir. Yes, sir. Sir, like, can you give the example of vertical gene transfer for like plasmid?
Plasmid is not vertical gene transfer; it is an horizontal gene transfer.
Sir, you said something like that about in last class.
Yeah, vertical gene transfer is that what you get from your mother to your daughter. That is vertical gene transfer because it is a part of the reproduction process. And horizontal gene transfer is that some genes jumps from one cell to another and another cell. It may be something which you have assisted or it may be a natural process. So that is horizontal gene transfer. That is not transformed from one progeny to another.
Okay, sir. So, like when we say that one RNA polymerase bind tightly to T7 promoter and not that tightly to lag promoter, what does it mean to us? Like how tightly, like they are forming bonds, something like that, or what?
There is a parameter called the binding constant. It depends on the value of the binding constant. If the binding constant is high, the binding is high. If the binding constant value of binding constant is low, that means the binding is not so strong.
That is that I can understand, but like on what it depends. Something like on chemical, there may be like forces also something. I was thinking.
Is something like a Lash-Hutelier equilibrium. How much of the fraction is bounded and how much is free. So if most of the fractions are bounded, that means the binding is strong. If most of the fractions are unbounded, then the binding is less. It's a thermodynamic property.
Okay, sir. Sir, again, like in last class, I saw that in one plasmid there were two ORI. So, like, why do we need in that small plasmid, like 4 to 5 kb plasmid, two ORI? Like in bacterial cell, large bacterial cell, there is only one ORI. Most of the cases, like, so why do we need two ORI in just 4 to 5 kb plasmid?
Who said in bacterial cell there is one ORI? There can be multiple. You can put as much ORI as you want. The whole idea is to have your plasmid making multiple copies in small amount of time, as much short amount of time as possible. If you add two ORIs than one, your plasmid will be able to make higher copy number because the DNA replication has started from both the points, and it will be faster rather than if you have started with a single point. That's why the bacterial that's why the human genome has multiple origin of replications, and they all start together. Even in bacteria this happens. The doubling time of E. coli is 20 minutes, but the replication time of E. coli is more than 20 minutes. That means always your chromosome is in a replicating state if the cell has to divide in 20 minutes.
But that all the replication bubble doesn't form in the single chromosome in that case.
How not?
Okay, sir. Okay.
Okay, then can I end up? Okay, I think most of the queries have been answered. So you prepare your lab reports and submit to KJM, and then I will leave. Okay, this class.
Thank you, sir.
Thank you, sir.
I just wanted to know a single question is that if anything you guys wanted to know and I haven't covered, which I can add to the next one.
Sir, just ask where to submit the report.
और यार ठीक है ओके फाइन ठीक है ओके बाय बाय